All right, there's going to be three things that we're going to talk about in today's lecture. The first thing is what is a DNA ladder? And we actually talked about them as standards when we ran our DNA gel just a little bit ago. The second thing is what is a PCR machine and how does it work? And number three is what are STRs and how are they used in the CSI lab. So I think all three of these things, are basic stuff from case one of that CSI lab, and it's what I'm going to quiz you on, so I wanted to be able to review it. So let's start with the first one. The first one is, what is a DNA ladder? And period 3 and 5 actually got to do a DNA ladder and make a standard curve and see that what it was, but people were absent there too. So I want to just kind of review. But basically, what a DNA ladder is that when we run our gels, in the first well, we run a sample that we know the size of the fragments that we put in. We know how many base pairs they are. And so, like maybe this one is 200 base pairs, this is 150 base pairs, this one's 100 base pairs, and this one's 50 base pairs. And so that way we can see when we run our samples, so A and B, we can say, okay, maybe this one and this one, fragment 1 and 2, run there. And then in B, maybe this one runs there and this one runs way down here. So what we are able to do is we're able to look at that and extrapolate looking at the standard. So what I mean by extrapolate looking at the standard, we can look at this one and say, okay, let me just get a little bigger here. We could say that one falls in between 200 and 150 base pairs on the standard. So if we want to look at that, we might want to say that like sample number one is, like 175 base pairs long. And we could do a graph and extrapolate like I did in class, but you could just eyeball it as well. So then let's look at these other ones. So sample number 2 is between 150 base pairs. So we could say sample number 2, keeps on doing that. Sample number 2 is, maybe 80 base pairs long. And then we could go and look at sample number 3. And sample number 3 looks a little bit bigger than 200. So we could say that maybe sample number 3 is like 215 base pairs long. And then sample number 4, if we look across, we see that it's less than 50. So I could say it's maybe 40 base pairs. Of course, a graph would be easier, and you can get a more closer number, but especially if you're using a computer like an Excel to make your graph. But this is a pretty good estimation, and so now you know how big the lengths are of the pieces that were cut by restriction enzymes. All right, so now we can look at the actual example in the CSI wildlife case, and you can see that we have two different elephants there, and you can see that, you can see that like fragment A is almost exactly at 180. Maybe it's a little bit bigger than 180. We might say like 185, but it's pretty darn close to that DNA ladder. Whereas B here is much bigger, and I would say that B is probably 190. It's between the 200. And so it's the same thing. And so you can go down through and you can obviously see how these two animals or these two elements have a different pattern. And their pattern is based on the STR, which we're going to talk about at the end. But there's definitely a fingerprint that shows up that shows them that they're individuals. Okay, so our next thing we're going to talk about is PCR, which stands for polymerase chain reaction. And what PCR is a technique which basically copies DNA over and over. A lot of times, in this elephant one, it was hard to get the DNA out of the bone, But even on a crime scene, they use PCR all the time because if you're if you just have a little bit of DNA left on skin that was during, like a murder or a little drop of blood, that might not be enough to run a gel electrophoresis fingerprint. So they want to make a whole bunch of copies. And so I like to think of PCR as just like a DNA copy machine. It was very interesting to me that it's based on the fact that when you have a double-stranded DNA, scientists realize that when you heat it up, and you have to heat it up pretty good, like if normally a body's at 37 degrees Celsius, but if you heat it up to 90 degrees Celsius, the two sides of the DNA will denature from each other, so they'll pull apart. Okay, and then you want to copy them when they're pulling apart. So on the next slide, you can see that. On this next slide, you can see kind of like the whole idea. So you have right here in step number 1, you have a piece of DNA and you have all the nucleotides and all the primers ready to go, but you heat it up so that it denatures into the two halves. And now you have it split into the two halves. Now, these primers, and also there's going to be what's called Taq polymerase, which is kind of like a DNA polymerase, but it's in a special organism. So Taq polymerase was a polymerase, like we talked about in replication, that was found in hot springs bacteria. And so that's what makes this possible, because we can we'll heat it to denature it, and then over here in step 2 we're going to add the primers, and they're gonna stick to the DNA, and then in step 3 we're going to add these Taq polymerases, and these Taq polymerases hit the primer and the DNA, and they run along and they copy the DNA. Now we're still at about 50 degrees Celsius. So you can't really, like our body's DNA and our DNA polymerase and our primers and everything would all denature at 50 degrees Celsius. But what makes this possible, PCR possible, is that we've got the polymerase, like the DNA polymerase called Taq polymerase, out of these bacteria, and we're utilizing their ability to survive in a very hot climate. And so the polymerase runs, and then boom, by step 3, you have 2 identical copies of the original DNA strand. Okay, so you ran that one time. So what a PCR machine does is a PCR machine is programmed. It's like a little computer that has all the nucleotides, all the primers, all the Taq polymerase, all the DNA. You just add your DNA sample that you wanna use, and it goes through this heat, denature, annealing or sticking the primer, having the Taq polymerase jump on, extending it. It does it over and over. It does these cycles. So this might be one whole cycle. And then it does it again, and it just keeps repeating. And if you just keep repeating that over and over, eventually you're going to get a lot of DNA. So like I said, it's like a little DNA copy machine that does it over and over. Okay, our last topic that I wanted to talk about, which are short tandem repeats. And I thought it was interesting when I was doing the CSI thing that they brought up short tandem repeats. But what they are is, it's not like when we're doing CSI, we run the whole elephant's genome, because that would be expensive and take a long time. And you guys have heard that, everybody is basically 99.9% the same. And same with elephants. They're probably almost exactly the same. So what they found is in these short tandem repeats, or STRs, is that there's these weird little areas that aren't genes at all. They're just like little segments of DNA. They don't code for anything, but are repeats. So down below here, this is a STR, and what's repeating here is GATA, GATA, GATA over and over. And let's see how many times. 1, 2, 3, 4, 5, 6, 7, 8. It repeats it 8 times. Okay. These random repeats, or these short tandem repeats, have on each side, they have what's called flanking sequences. And this is like where we know we're gonna have these repeating segments. Now, so what's kind of interesting about these is that they're very, very, very unique to each individual, or they're variable. So every elephant can have a different length, although they'll all have the GATA at the same spot. You know, another elephant might have it only repeating 5 times. Okay, and then also, you know, DNA comes, you get one from your mother and one from their father. So like the father's DNA could be like GATA 10 times, and the one that you got from your mother might be GATA repeated 5 times. So you're going to be, in that case, you'd be heterozygous. All right. So now some individuals, maybe they get the same exact one from the mother and the father. So they're going to get the GATA repeat, and it's going to be 5 times. And then from the mother, they're also GATA repeat is going to be 5 times. And these people would be homozygous for that specific STR. Okay, so why is that important? So when we run a gel, since this is, that's not doing what I want. Since this is, we're going to go ahead and do this. When we run a gel, some elephants could show a homozygous. The band would come up at the same time because they're the exact same lengths, maybe 5 base repeats lengths. But this guy is heterozygous, and he's going to show 2 bands. So, of course, you can't identify an elephant just by one STR, because most of them are going to fall into a range of like 5 to 6 to 7 to 8 or whatever. They're going to be kind of in a certain area, and there's quite possibly could be multiple elephants in the same population that share the same length and either heterozygous or homozygous. But scientists, in our case, in what we've been looking at, scientists would compare, I think it said 16 different STR markers. And these markers, when you have 16 of them, the chances of having differences amounted, and 2 elephants having all the same differences and coming out exactly the same is like 1 in so many billion that it's more than the elephants in the world. So that's physically impossible to happen. So if you run enough of these markers, it's very unlikely that any 2 individuals will be the same. And scientists calculated that 16 markers is the amount that there's no 2 individuals that would be the same. So again, STR stands for short tandem repeats. They're highly variable, meaning that each individual can have a unique allele, and an individual can have 2 different alleles, thus being heterozygous. Okay, that's it.